In this video, we are going to explore the development and analysis of amino acids and nucleotides in order to create a molecule that would seek out, hunt down, and kill coronavirus. Coronavirus's genome is an RNA genome. It is approximately 29,900 nucleotides in length. The nucleotides are basically genetic code strung together. The RNAs use 4 different nucleotides. These nucleotides are A, C, G, and U, which stands for adenine, cytosine, guanine, and uracil. The DNA uses 4 nucleotides. The DNA's nucleotides are A, C, G, and T for adenine, cytosine, guanine, and thymine. There are transcription factors. Specifically in this discussion, we are going to talk about transcription factor 3A, and we're going to talk about modifying transcription factor 3A into a molecular virus killer. Transcription factor 3A is used as the anchoring molecule of a much larger complex called the transcription complex. Transcription complexes are comprised of a number of transcription factors, in some cases many as 72, and these transcription complexes are used to decode the genetic information which is stored in the DNA. The role of the transcription factor 3A molecule is to bind to the DNA. It uses zinc fingers 1, 2, 3, 4, and 5 in order to accomplish this. It binds to approximately 30 different nucleotides in the DNA, a specific sequence of the DNA. Once this occurs, then zinc fingers 6, 7, 8, and 9 are used for other transcription factors to come and bind to this particular one, and from there, a much larger complex of transcription factors is created. And then the entire transcription factor complex then moves down the DNA, reads the genetic information in the DNA, and then creates RNAs. There are approximately 20 different amino acids which are used by the body to create the 33,000 differing proteins that humans need. Transcription factor 3A is comprised of 365 different amino acids. That is, amino acids strung together. There are 20 different amino acids, but this combination creates this particular form of a molecule compared to the other 33,000 different proteins that are used by the body. The problem that faces us is being able to take this particular molecule, modify it such that this particular molecule will seek out a 30 nucleotide sequence of COVID-19's genome. Again, as we discussed, COVID-19 has 29,900 nucleotides which comprise its genome. So we are looking for a unique sequence in that 29,000, almost 30,000 nucleotides for this particular molecule to seek out and attach to. Now, the key is that if you find such a sequence, then how do you bind a molecule like this to that genetic material? And my father, Dr. Lane Schreiber, Sr., was able to use spatial analysis and bonding analysis in order to look at how, in 3-dimensional space, these amino acids would make contact and bind to nucleotides. The DNA is a double helix, and in space, it twists in space. And so therefore, then, you need to account for that. Again, the COVID-19 is an RNA, slightly different problem. But having said that, with regards to the 20 different amino acids which are used in order to construct proteins, my father was able to determine that asparagine, which is N, which is an amino acid, binds tightly to adenine, one of the nucleotides. Glutamic acid, which is E, is an amino acid which binds tightly to cytosine. arginine, which is R, binds tightly to guanine, and lysine, which is represented by K, binds tightly to thymine. This is the key, because with this binding scheme, we are able to code out the amino acids on transcription factor 3A in order to be able to use this molecule to bind to COVID-19's genome. And in the process of that, if we're able to accomplish that, then this 365 string of amino acids will bind to COVID-19 and prevent COVID-19 from being able to replicate. And if it can't replicate, the infectious process has been ground to a halt. What we need is the assistance of engineers, programmers, biochemists, bioscientists, the general public who's interested in using their imagination and their analysis skills to be able to determine the most optimal sequence in COVID-19's genome for us to be able to use a molecule like this to hunt down and kill coronavirus. These are difficult times. This is a very wicked virus. My friends, be careful, be safe.